was a call of an echolocating Myotis natereri bat, using its call to gain an understanding of its surroundings and pursue prey. Echolocation is the use of a biological sonar to assess the environment for navigation, foraging, or hunting when visibility is compromised. Many bat species have a highly evolved auditory system to process the call and returning echoes and extract information about their environment and prey. How does the bat convert call and echo auditory information into a sophisticated representation of its surrounding environment? And how do they use this information to generate a cortical representation? The neural processing required to use the system to navigate their environment is complicated and intricate, and in this presentation we will identify the main pathways used in forming a cortical representation and the neural processing involved to achieve this. This is a spectrogram, a visual representation of frequency spectra for a given signal over time. This spectrogram shows typical bat echolocation calls. Calls consist of a number of harmonics, including the fundamental, usually between 20 and 40 kHz, and the subsequent harmonics, including as many as 3. Generally, the most common signal type in echolocating bats is the frequency modulated call, where the frequency is modulated as the bat is searching for prey. However, many bat species, including the horseshoe bat and the mustache bat, are bisonar and use a CF-FM call composed of a constant frequency component, a sine wave, and a modulated frequency component, which involves a sweeping reduction in frequency. Generally, the CF component is used when the bat is flying, and the FM component when it has identified a target. A cortical representation refers to how the bat represents sensory information about its environment in the cerebral cortex. Generally, auditory representations form maps on the cortical surface, where localization of activity is relevant to some environmental feature. This kind of setup is referred to as computational anatomy, and is a common theme for many cortical representations of different forms of sensory information. The mustache bat, Pteronotus parnelli, is a model organism often studied for bat echolocation. This bat species has a bisonar call with a CF and FM component. The bat auditory system is like that of other mammals, but is highly adapted. There are particularly enlarged inferior colliculus and auditory cortex, as these areas need a lot of neurons devoted to the processing of sound. There are particularly large regions dedicated to the processing of the fundamental and the harmonics of their call. The textbook auditory pathway, shown here by the grey arrows, is an ascending pathway. Sound waves disturb hair cells in the cochlea of the bat's inner ear, which in turn causes auditory nerve fibres to discharge. They first synapse in the cochlear nucleus. Beyond here, nerve fibres ascend to the superior olive in the pons, and also in the nuclei of the lateral lemniscus, leading up to the inferior colliculus in the midbrain. Next, nerve fibres ascend towards the medial geniculate nucleus in the thalamus, and then onto the auditory cortex. As you can see, however, there are multiple routes information can take, and even descending pathways. This is an example of parallel pathways, and all seem to be necessary for echolocation processing. There is evidence that along these pathways there is integration and transformation of signals. Timing in the bat's auditory system is really important for accurate echolocation. The neural processing involved in echolocation relies on the bat's ability to detect time delays between the initial call and the returning echo. From this, the bat can understand the target range, the object size, and the target's position relative to its own. There are mechanisms in place to transform the temporal properties of a signal at multiple levels. This is important for neural tuning, or making neurons only respond to specific sound or signal properties. Transformations of these signals must occur such that the information can be represented in the cortex. There are 3 main mechanisms. Firstly, neural lateral inhibition alters the temporal properties of response patterns. Glycine and GABA are the main neuron types associated with this activity. There seem to be a lot of inhibitory inputs from the nuclei of the lateral lemniscus. Neural inhibition may decrease the properties of certain neurons when another one becomes active, which refines the signal and allows for many other properties. Secondly, discharge patterns are transformed in the nuclei of the lateral lemniscus either by their onset or duration. Cells in the columnar division of the ventral nuclei of the lateral lemniscus show no spontaneous activity and respond with only one spike, shown here on the left, correlated with sound onset. Despite auditory neurons displaying sustained firing, transient responses may be created by the convergence of 2 sustained inputs, one being excitatory and another being inhibitory, which alters the transient onset and offset. Responses from the rest of the nuclei of the lateral lemniscus are sustained throughout the signal duration to convey information about the length of a sound. Combining excitatory and inhibitory input tunes for simple temporal features of a sound. Finally, delay lines are present in the nuclei of the lateral lemniscus and in the inferior colliculus. These increase latencies in responses. Delay line systems can increase latencies by a few mechanisms, two of which are particularly seen in the bat auditory system. Longer neurons simply increase the distance the impulse has to travel, which therefore increases its latency, and narrower neurons transmit impulses much more slowly than fatter ones. Secondly, the presence of synapses in interneurons can increase the latency as synaptic transmission is slower than axonal transmission. These pairs of neurons are connected to the same neuron, which will fire when the impulses arrive simultaneously. This type of neuron is known as a combination neuron or a coincidence detector, and is perhaps the most important feature of the echolocation system. Furthermore, there is convergence and integration in the inferior colliculus to tune neurons. Firstly, neurons can be tuned for sound duration, particularly in bat species that vary the length of their call, like the big brown bat. This tuning is likely achieved by neural inhibition and excitatory mechanisms. In this example, the neuron is tuned to sound durations of 5 milliseconds. A shows the coincidence of a depolarization from a lower neuron stimulated by the sound onset and the depolarization from a lower neuron stimulated by the sound offset. When the sound persists over 30 milliseconds, shown in B, the depolarizations do not coincide and do not produce action potentials in the duration-tuned neuron. Secondly, neurons can be tuned to frequency sweeps in the FM component, termed FM specialists. There are a few models to suggest how this is achieved in the inferior colliculus, but each of them suggests that the neurons are selective to the temporal features of the sweep and the direction, which is determined again by the coincidence of depolarizations. Thirdly, neurons can be tuned for modulation rate of frequency or amplitude. Certain neurons in the inferior colliculus respond selectively to specific rates of modulation. For instance, cells can be selective to sinusoidally frequency modulated tones, usually at low modulation rates. Finally, delay tuning involves the tuning of neurons to the call echo delays. This is by the use of coincidence detectors and delay lines. We shall see that tuning to delay is perhaps the most important neural processing mechanism in echolocation, and is used to assess how far away a target is. The pursuit of prey requires a highly efficient and accurate echolocation system, as the bat must make very quick and sudden movements to keep up with the fleeing prey. In the next section, we will look at the subcortical and cortical processing involved in generating cortical maps that the bat uses when it's pursuing prey. The two most important characteristics to establish are the target range and the approach velocity. Target range is computed using call echo delay information, and approach velocity is established using a Doppler shift that changes the echo frequency. As the bat approaches a target, the returning echo undergoes the Doppler effect and gets shifted up in frequency. This change in frequency is processed in specific regions of the auditory cortex to compute approach velocity. The time delay between the call and the echo is also computed in the auditory cortex, which is used to establish target range. So, how does the bat detect these changes in frequency and the time delay subcortically? The first stage of this clever processing is at the basilar membrane in the cochlea of the inner ear. In the cochlea of the mustache bat, there are approximately 18,000 hair cells on the basilar membrane that detect sounds of specific frequencies. Each hair cell is tuned to a best or characteristic frequency to such a high degree that if its characteristic frequency is 60 kHz, a deviation as much as 1 kHz can result in a drastic reduction in stimulation. As you can see in this diagram, the characteristic frequency can stimulate the neuron at very low intensities, indicating the neuron can respond best to this frequency. This creates a neural code for the frequency of sound along the basilar membrane. The tuning curves for the fundamental frequency and the harmonics are the narrowest of all, indicating they are the most precise, and bats have evolved highly precise auditory nerve fibers for the purpose of echolocation. This tonotopic organization of the basilar membrane is an example of computational anatomy. Remember that the mustache bat is bisonar, and thus has a CF and FM component of its call. Neurons are also tuned to this. CF neurons respond best to the CF components of the call and the echo, whereas FM neurons respond best to the FM component. This tuning is achieved by the transformation and integration mechanisms that we discussed before. The 30,000 auditory nerve fibers must then project to the cochlear nucleus and onto the inferior colliculus. Bats have a large inferior colliculus to begin processing all this auditory information, and in mustache bats, the 60 kHz processing region is the largest as it is the most energetic harmonic of the call. In the cortex, it is not as simple as simply having tonotopic organization. Sensory modalities in the cortex have multiple maps and representations of environmental information received. Coincidence detectors play an important role in the processing of call echo delays and the determination of target distance. These neurons are in the cortex and are converged upon by lower-level auditory nerve fibers and are either CF-CF or FM-FM, meaning they respond to either a combination of CF neurons or FM neurons. These form the FM-FM area and the CF-CF area, to name two sensory modality areas. There is also the Doppler-shifted CF area that is involved in the fixing of the problem of the Doppler effect. Coincidence detectors in the FM-FM area can detect the simultaneous arrival of inputs from these combinations of neurons. Remember that the FM1 call of the mustache bat is particularly weak, meaning the echo is even weaker, so the call itself stimulates the FM1 neurons. The harmonics of the returning echoes stimulate the FM2, FM3, or FM4 neurons. The coincident arrival of these stimulated inputs causes FM-FM combination neurons in this area to activate. This is how echo delay works. The FM-FM neurons are also localized depending on a specific temporal order, i.e. the time delay. The location of FM-FM neuron activation is an important step to determine the target range. There are two dimensions of this area: the combination of the FM1 component to its harmonic relating to target distance and the frequency of that matched harmonic. Temporal order is represented along the x-axis and the frequency of the combinations on the y. The more rostral regions are for shorter echo delays, whereas larger distances are more caudal. So, if there is a large call echo delay indicating a greater target range, the FM-FM combination neurons that are more caudal are activated. As a bat closes in on its prey, the activity moves caudal rostral along the FM area, and when the target range is small, the most rostral FM-FM combination neurons are active. For reasons explained later, the y-axis also represents slightly different frequencies of FM1. As the 60 kHz component is the most energetic in the mustache bat, a large portion of the FM-FM area is dedicated to processing FM1 and FM2 combinations. This computational anatomy explanation suggests how an approaching target is represented in the brain of the mustache bat. But if the call and echo are separated by a delay, how do they arrive at the combination neurons at the same time? The answer is delay lines. These increase the amount of time a signal takes to reach the coincidence detector. So, when a delay line delays a call-stimulated signal to such an extent that it arrives at the coincidence detector at the same time as an echo-stimulated signal, this gives information about the distance in the form of computational anatomy. Delay lines and the transformation and integration mechanisms allow temporal tuning of the FM-FM area. As the bat approaches its target, the Doppler shift means that the frequency of the returning echo is shifted, sometimes as much as 1 kHz. This means it would not be detected by the highly tuned neurons due to their narrow tuning curves. Does the bat solve this by having neurons tuned to every possible frequency of the harmonics and fundamental? No, there simply isn't enough room to have that many neurons. The problem is solved by adjusting the frequency of the call to compensate for the Doppler shift, so that the returning echo is in the range of the filter neurons. This is Doppler compensation. This only occurs when the bat is approaching the target, as this kind of precision is only necessary when the bat has detected its prey. The Doppler shifted CF area contains isofrequency contours to represent neural activity responding to the different Doppler shifts. As the bat receives a CF2 echo component of a certain frequency, let's say 61 kHz, i.e. the resting frequency when there is no Doppler shift, there is activation of the 61 kHz neurons that are arranged concentrically. When the CF2 echo component is 62 kHz, i.e. when it has been Doppler shifted, there is activation in the 62 kHz neurons in this area. This area also computes the intensity of a stimulus, and these are arranged radially. The DSCF area can detect frequencies between 60.6 kHz and 62.3 kHz. This then gives feedback to other brain regions so that the bat can adjust the CF2 frequency of its outgoing call so that it returns at the resting frequency. This system is great at keeping the CF2 component frequency relative and constant. As we shall see, the CF-CF area has mostly combination neurons responding to CF2 neurons. Only the second harmonic undergoes Doppler compensation. This is because it is the most energetic and most relied on to assess target range, so any issues in computing the returning echo could have problems with the bat computing target range. All other harmonics must be tuned to the different fundamental frequencies of the call, which depends on the velocity. In a perfect system, everything would be Doppler compensated. However, there is only so much energy that can be put into neural circuitry, so evolution has found the most efficient and least expensive way to compensate. The Doppler shift compensation keeps the frequencies within the acoustic fovea for the CF2 components. For the FM components, the frequency of the Doppler shifted echoes still falls within their range of auditory responsiveness, as seen in the FM-FM area. As we saw before, there is tuning to slightly different frequencies of the fundamental and harmonics in the FM-FM area, and it is for this reason. Doppler shifts are also used to compute the flying velocity of a bat. This is seen in the CF-CF area, which computes rate of approach and creates a neural map of approach velocity. The frequency of a call and of the echo could be compared to assess this. Combination neurons in the CF-CF area match the frequency of a call with that of the echo by computing Doppler shifts in the constant frequency component of the signal in the echo. In the CF-CF area, neurons that respond to the same frequency of the CF1 call component are arranged in axial ribbons. Combination neurons tuned to the same Doppler shift are also arranged in axial ribbons. The locus of activity within the area changes as the approach velocity changes. CF1-CF2 combination neurons are the most abundant in this region. In the more caudal region, neurons are tuned to higher frequencies of the CF1 call component, and activity moves more rostrally as frequency of the CF1 call component lowers to compensate for the increase in frequency caused by faster velocity. Thus, the fastest velocities are represented rostrally. This is represented much like the computational anatomy in the FM-FM area, two-dimensional with one dimension representing velocity and the other representing the matched frequencies. Neurons respond best to high CF1 frequencies are located more rostrally. A specific Doppler shift is represented as a spatial location on the map. Remember that the Doppler compensation system cannot predict the future and only responds cyclically to a Doppler shift, so it's not 100% accurate, but it is accurate enough. Again, the subcortical transformations and integration mechanisms allow the tuning of this area to the velocity. Thus, the bat can compute the target range and the approach velocity. Is this enough cortical representation to be able to navigate the environment and hunt successfully? Not quite. There are potentially more than 10 neural maps and representations of prey pursuit in the cortex. Binaural and monaural time differences are crucial to localize objects in the horizontal and vertical planes. However, the areas we discussed are unique to echolocating bats. There are even more regions to deal with problems during echolocation, including compensation mechanisms for the changes in call and echo frequency as a result of air disturbances from wing beats. Now that we've established the basic cortical representations allowed by ascending pathways, we can look at the role of descending projections, often termed corticofugal, literally meaning running away from the cortex. We can ask the question, what corticofugal or feedback pathways are in action to modulate or improve the ascending pathways involved in echolocation? Recall that frequency tuning curves are sharp, particularly for the fundamental frequency and the harmonics. This is achieved by lateral inhibition mechanisms from projections ascending between regions. However, inactivation of the corticofugal pathway by studies has shown its importance in the fine-tuning of neurons, a feature which is highly important for creating a cortical representation with high acuity and precision. Many studies have studied the role of the corticofugal pathway by inactivating it. Lidocaine specifically inactivates cortical neurons tuned to frequencies between 60 and 65 kHz. This study by Zhang, Suga, and Yang investigated the responses of thalamic and collicular neurons following this cortical inactivation. Cortical inactivation caused by lidocaine decreased auditory responses of the matched thalamic and collicular or subcortical neurons. However, this did not change their tuning curves. They remained responsive to the same characteristic frequency, but they required a greater stimulus. Conversely, cortical inactivation increased the auditory responses of unmatched subcortical neurons and shifted the best frequency towards the inactivated cortical neurons. Thalamic neurons showed a larger change in response magnitude than collicular neurons for both matched and unmatched neurons, indicating they were more greatly affected by the cortical inactivation. So, after cortical inactivation, only unmatched subcortical neurons' best frequencies shifted, and matched subcortical neurons' best frequencies did not shift at all. Also, the decrease or increase in response and the shift in frequency tuning of the subcortical neurons occur both in the colliculus and the thalamus through the corticocollicular and corticothalamic projections, but to different extents. What does this say about the role of the corticofugal system? If when we inactivate regions of the cortex there is a decrease in matched subcortical neuron response and an increase in unmatched subcortical neurons response with a shift towards the best frequency of the matched ones, we can conclude that the opposite happens when the cortex region is active. We can conclude that active cortical neurons augment responses of matched subcortical neurons but suppress those of unmatched subcortical neurons and shift their characteristic frequency away from those of the original cortical neurons. This is termed egocentric selection and is a result of focused positive feedback and lateral inhibition mechanisms. This selection achieves multiple things. First of all, greater acuity and finer tuning, and furthermore, egocentric selection is a fundamental function in the auditory cortex and is involved in echo delay processing. When an identical signal is received repeatedly, egocentric selection will increase the neural representation of this frequency occurring in the colliculus, the thalamus, and ultimately in the cortex. Neural representation is decreased if a signal is received rarely. It is likely that egocentric selection is involved in long-term changes in the overall functional organization of the auditory system, particularly in the frequency tuning, which as we know is important for processing call echo delays. This leads to a well-known phenomena of brains to change their structure and physiology depending on environmental cues, plasticity. Plasticity is important as it ensures that the correct amount of processing and neural space is given to the most relevant brain regions and for the most relevant stimuli. The shifting of frequency tuning in the cortex is an example. A further study by Gao and Suga has suggested a positive feedback loop involving ascending and descending pathways in the auditory system of echolocating bats to improve cortical neuron responses to specific frequencies. This study found a positive feedback loop in the neural circuits of the big brown bat, another species of echolocating bat that has just an FM call. Sound conditioning can change response properties of neurons in a sensory map. A constant tone can induce a shift in the characteristic frequency of subcortical neurons and cortical neurons. This suggests an ascending pathway of augmentation of responses as the subcortical neurons shift their characteristic frequency in response to the conditioning stimulus, which then shifts those in the cortex. However, focal electrical stimulation of the auditory cortex evokes collicular and cortical characteristic frequency changes that seem to be the same as those evoked by conditioning stimuli. This suggests that the cortical electrical stimulation is inducing a change in the frequency tuning of corticofugal neurons via the corticofugal pathway. So, the response properties of neurons and the frequency mapping in the cortex can be altered by auditory conditioning and focal electric stimulation, suggesting both ascending and descending pathways are involved in plasticity. Gao and Suga hypothesized that cortical activation adjusts and improves subcortical neuron response via the corticofugal pathway, which can then boost cortical characteristic frequency shifts by ascending pathways, a positive feedback loop. Otherwise, the cortical characteristic frequency shifts would be fairly small. This process allows greater plasticity. However, other brain regions have a say in some of this processing. There are also connections from non-auditory brain regions that determine the relevance of acoustic stimuli. These regions, such as the amygdala and the forebrain, augment cortical changes depending on the behavioral relevance of them and can boost cortical acetylcholine levels to increase the cortical characteristic frequency shifts. For instance, the repetitive sound of an insect is more behaviorally relevant than a rumble of thunder in the distance. So, the cortex and its maps and environmental representations are plastic, depending on the characteristics of the sound and the behavioral relevance. So, ascending and descending pathways have a role in cortical representation as signal processing. Taken together, these findings show the importance of ascending and descending connections between subcortical and cortical regions to create environmental representations in the cortex. Spectral and temporal features from calls and echoes are transmitted via ascending pathways from the cochlear nucleus up through to the inferior colliculus and eventually to the cortex. Along these ascending pathways, signals are transformed and integrated to tune neurons to specific sound characteristics, such as frequency, delay, and duration. In the cortex, there are specific regions to compute target distance and approach velocity by creating maps. This is a form of computational anatomy. There are also clever Doppler compensation mechanisms that recognize a change in signal frequency due to Doppler shifts and compensate for this accordingly. Studies investigating the descending pathways in the mustache bat and the big brown bat species have alluded to its role in the positive feedback mechanisms to allow greater tuning of neurons, and also in plasticity. All of these mechanisms together make echolocation in bat species possible. Clearly, years and years of evolution have led to highly adapted and sophisticated computation systems. Research is still being done today into the exact mechanisms involved, with huge contributions from the researcher Nobuo Suga at Washington University. Also, beyond just being interesting, knowledge of bat echolocation has real-world applications. Sonar heads that mimic bat echolocation have been developed, which involves an array of receivers and ultrasonic emitters to mimic the signal pattern of echolocating bats. Furthermore, assistive technology for the blind are being developed. These handheld devices emit ultrasonic pulse echoes to determine object distance. Also, bat echolocation behavior inspired the creation of the BAT algorithm, a meta-heuristic algorithm that addresses global optimization engineering tasks, or tasks of finding the best set of admissible conditions to achieve an objective as a part of nonlinear programming. BAT echolocation behavior can be formulated to be associated with an objective that needs to be optimized, such that new optimization algorithms can be formulated. With all this exciting real-world application arising from computational understanding of bat echolocation, who knows what may be developed in the future? Echolocation is really a fascinating behavior.